a faculty presentation. Our faculty program is where professors come in and work with scientists and collaborate on projects over 3 summers. So we have a mixture of some that have just started this summer, and then we've had some that's been with us for some years, and we're very grateful for them. So today we have 3 presenters, and we're going to go ahead and let our first presenter get started. Dr. Nhan.
Yeah, thank you so much, Nikia. So such a great pleasure to kick off the day. So let me share my screen. Yes. All right. So so you see the title of my my talk is robust and efficient rank structure solvers and reconditioners for singly perturbed differential equations. So my work this is joint work with Yang Liu and Peter Geisos from the group soluble solvers group in math, applied math division. And because I only have 10 minutes, so I should do everything quickly. So let me begin. So here is the outline. I have 10 minutes, but on 7 items, so it's a lot. But it basically is the question, the items that I pick up from Nikia's list, so that to just focus on the things that I want to talk about. So first, I would like to make announcements, and my thanks go to Sherry. She's here today. I'm very lucky to have her, and I met Sherry in 2019 in a conference, and at that time Sherry invited me to give a talk, research talk to the group, and then I make the connection with Yang and Peter there, and then this is a great chance for us to continue to have a common interest, and then we write a project, a proposal, and then we got the funding. And I also want to acknowledge my collaborator and my PhD supervisor, Neil Maiden from Ireland. And I also want to thank the team, especially Colette and Kia and Ingrid, on the people that makes this happen and easy for me to jump in and do the research. So here is about Hollingham's, my university. So Hollingham is very close to the lab, the neighboring city, like Jim, and it's we in the Oakland Hill. So you have the beautiful bay view like from the lab, and this is a very small university, and it's founded in 1868. 6 sisters comes from Quebec to the Bay Area, and they settled out. And the student population is very small. We have about 600 undergrads and about 300 grad students. But we don't have math majors. We have majors in biology, psychology, business, and music, and something like that, but not in mathematics. So that's briefly about my university. And let me talk about the differential equation that I'm interested in. And the application here, you can see the volcano, you see the ash bloom. We usually using this differential equation when you have combination of the processes. Here, for example, you will see on the picture on the left, you will see the diffusion process. That means the ash bloom spreading out, right? And the convection process, that means the flow of the bloom, right? But here the flow convection term will be dominated. Why? Because you have the source, the volcano is operating strongly, right? So that's the processes. Another picture you can see, when you put a dye into a flowing river, you have the diffusion, and you have the convection as well. And you also have the reaction diffusion term when you have the when they are singly perturbed. That means when you have some reaction between two things, like you have rabbits and then carrots, you see they interact with each other, and they have the diffusion, which is spreading out of the carrot, or the spread of the rabbit. So that's the application of my equation, and put that in complex setting, and now I put everything in very smaller scale, similar scale. Look, the reaction diffusion on the top and the convection diffusion on the bottom. But the thing that I want you to focus on is the very small parameter that multiply into the diffusion term here, you see. And we are interested in the case that the parameter, the singular perturbation parameter here, getting very, very small, close to zero, right? So that's the case. Now I want to show you some pictures, some solutions here. Look at the epsilon equal to 1, right? Look at what the behavior of the solution. When I let the epsilon goes to getting smaller, let's see what happened, right? So you see, this is the changes when I put the epsilon to 10 to the -1, 10 to the -2, you will see that the solution changing, and there's boundary layers, say up here, occurs here in the graph. You see 4 layers on the left, and then 2 layers on the right for convection diffusion. Getting smaller, you see, when epsilon equal to 10 to the -4, you see that you have a strong boundary layers at the boundary regions right here, right? So that's, so what are the 5 minutes. Okay, so what are the challenging here? Sorry, how do I go back? Yeah, so the steps, when you have 2 steps, step 1 theoretical work, and then the step number 2 is the computational work. And mainly the challenging task is solving the large linear system coming from the discretization. Yeah, look at the time when you saw here. When you have the parameter, the discretization parameter n increases, you will see the increased time, of course, by the complexity of the system. But now there's another thing you look at. When you fix one n, you will see the number, the time increase when epsilon is not uniform in epsilon. So the goal is, can we do better than that, which is using the Chomod, the state-of-the-art direct solver on the market there. So my question of the project is, can we improve that, right? And now another challenging part is dealing with the subnormal numbers, because if you see the subnormal number is just something very small that the hardware or the compiler cannot officially calculate. So we need to something that we need to do on the microcorp at another level. It's just more expensive in terms of the computational cost. So look at this is the Cholesky factorization of the matrix. You will see when epsilon equal to 1, you will see I have 2 minutes. So let me go. All right, let me skip that. Just now, the method and material we employed Chompak, which is developed by the group, the lab group, and then we use the supercomputer. So let me show some result. Skip this one. Now, Cholesky factorization, if I grab this one here on my computer, i5 2.4 gig processor, and you see the time significantly improved in terms of using the Chompak, which is around 10 seconds to solve these things. But now another thing is, when that's for a system of about 1 million unknowns, And when I increase to a system of nearly to 17 million unknowns, you will see the time, the factorization's time, the exact solver here is just about 55 seconds. And then with low rank compression, we have just the factorization time is just about 10 seconds. But again, there's a associate, there is a inconsistency in terms of the epsilon, the values of epsilon. Okay. So now this is so we plot with the supercomputer at the lab. We are able to increase the number of unknowns up to about 67 million unknowns, and we are able to test the time, time for the multifontal method with the compression right there, and you see the scale here. Which is the linear scale, which is very good, linear scale. And then the multifactor exact solver doesn't work when with the 67 million unknowns right there. So that works very well for us. And the outcome, so the outcomes, I list a few outcomes here. The first time we apply multi-solver under the Trump package to solve the problem, and we used the supercomputer from the lab, and the initial experience, as you can see, that the low rank approximation with the multifontal method works very well. And we also add the new feature into Trump. We are able to count the subnormal numbers appear in the factors of the multifontal as well, which is that's help from Peter, which is great. That we can looking at the factors a little bit clearer to explain the things. So for future work, we want to set other harder standard for us to solve 3D problems, for example, and other some theory work, the question that we need to answer as well. So that's the experience and story. So I enjoy and working with this is the first time I work in the research focused environment, because I'm coming from a teaching focused institute. So this is just a great experience for me. And this is for me to put my hand on, cutting research topics and state-of-the-art facilities from the lab. I don't have any students, so but I would tell my students that don't be discouraged when you study at the small colleges like this, and then when you have the opportunity, try to engage and take the opportunity. You can be part of the team. So conclusion slide. So my research about is just solving the very large linear system with application in other science areas, and how will my research impact the others? Yes, so that you can see that. So I think that's thumbs up for me, and thank you very much for your attention, everyone. Thank you. Thank you.
Does anyone have any questions for Dr. Nhan? You can either raise your hand or you can put them in the chat.
Hi, Anne. I just have a quick question. You said you use Cori. What kind of what kind of a submit job do you usually do? How many nodes?
Yeah, that's very interesting question. Hello. I did not have time to put my task, this is the first time set up to run Cori. I would say that start will be next visit. But I would ask Nhan, are you still here? Nhan is helping me running the Cori task there.
Right. Yeah. I mean, due to the time limitation, actually, yeah, we set up everything for Sai on Cori, and basically he provided us the example and the driver to generate the metrics, and then we can use whatever we have right now. So it's an easy integration process.
So do you think if you switch to perlmutter with GPU, it will help?
Yeah, I mean, Peter can answer this question better, but yeah, I mean, for the solver options I tried, yeah, they both have, I mean, both the exact solver and the Lorentz solver, they both have GPU capabilities. So we should be able to see better speed up on perlmutter.
Yeah, I just want to echo one of the challenging when I start is that I have the code in MATLAB and then the matrix, in different settings. So how can we, have everything, connected? And that was my first, fear when I jump into the project, whether I can, get over that difficulty to get things runs. Yeah.
Okay, I have another question. What other applications do you have for this model besides, you showed the volcanic ash coming out and the color that is spreading in water? Like, do you have any other applications for this model?
Yes, Sarah. So the application, yes, is coming from, for example, when you have the semiconductor equation, yeah, when you have a very small, something small in the diffusion, the first parameter of the, I don't remember the specific, but it's semiconductor. And then another thing is when you, for example, the Navier-Stokes equation, when you, after you linearize the equation, you will get type of this equation, because when the Reynolds number is very large, right? So this you have a very small parameter for the diffusion term, yeah. And yeah, for bio, it comes from bio as well. As I said, the reaction equations, diffusion equations coming from, so you have, say, particles interact with each other. Yeah, so that's the things that from my applications.
Nice, thank you. The electron, so the Navier-Stokes equations, like, the electrons in graphene, they actually follow Navier-Stokes equation at a certain density. So it seems like there is application on that,
Yeah, that's right. Right. Yeah. And the thing is, we want to solve the I don't have time to, in talk, but we want to solve them with any small parameter right there, not just, sometimes you just fix the parameter multiply into the diffusion, but we want to, find a method that robust in terms of the parameter right there. Yes, so that's the things. Yeah.
Thank you.
Yeah, thanks.
Thank you very much, Dr. Nhan.
Thank you.
Our next presenter will be Michael Cohen.
It's all right. That's a new one. All right, let me get rid of this. Oh, we just skipped a slide. Okay. Well, I'll take a break from fluid dynamics, and this I think is something I think everybody here. This will be accessible. What we have done this semester in collaboration with my mentor at LBL, Trent Northern, is to do some preliminary investigations just to see, do we have a phenomenon? And I should tell you that this is a completely virtual project, meaning it wasn't done at Lawrence Berkeley National Lab. It was done at Sonoma State, all the work that's described here. And if we do see something, then we can pursue it at LBL. And then I'm happy to say that there is something to pursue. So let me describe the nature of the problem in a second. But first, oh my goodness, what was that? I think I got this down. quick thanks. I'm not going to mention everybody here. There are a couple people who are on. So, I appreciate, especially this being virtual. It's required a lot of work on the part of the people in administrative office. Nakia is here and Ingrid's here. So I'll thank you right now. Everybody else, well, it's a short talk. So we got to move on. I don't know why it's doing that, but here we go. Okay, the nature of the problem. Anyone who's driven down 101 going to Los Angeles probably passed by several strawberry fields. And this is done with just berry cultivation in general, but many other crops using what's called plastic mulch. Mostly it's polyethylene, and it conserves water among other things. But one thing I realized with one of the people you saw there in the acknowledgement slide, from my contact at the UC Cooperative Extension in Watsonville, is that this plastic is basically just harvested every year, baled, and then it has a couple different fates depending on where it is, but it just becomes part of the problem, plastic waste problem, and it's not a minor problem, but it's easy to make plastic. You can't really recycle it any known means because the amount of soil that's with it, it's just very cumbersome recycling process. So what we wanted to do was enable the breakdown of these biodegradable alternatives, especially in colder climates like along the California coast, Washington State. It doesn't, these polymers, although they're biodegradable, they don't break down fast enough to keep them from accumulating in colder soils. And just a little bit here, polylactic acid, that's just a polymerized lactic acid, same thing you get in yogurt from lactic acid fermentation, and this polybutylene apatate tetraphthalate, PBAT, we'll just say, is a product of petroleum, but it's biodegradable. So it's not completely biological derived. The exact nature of this stuff, it's a proprietary mix made by the BASF of Germany. Now, real quickly, the experimental procedure, this the main experiment I did is taking weathered mulch, replicating what you would have under field conditions that you would be tilling into the soil, putting it into containers, the first 300 mils of soil, then upping it later, and with and without germinated seeds. And you can see all the conditions we tested. I'm not going to get into them, all of them, other than it's, we have soil that had previous exposure to polylactic acid, had been incorporated into plants, barley, switchgrass, corn. There's a reason for that is, well, they're known to put out proteases, which are the enzymes that break down polylactic acid. Other esterase enzymes break down the PBAT. But another treatment we did, and this is the one I'm going to focus on because it gave the positive results, significant positive results, is a spray pre-treatment of the mulch sections with a microbial community that we happen to have going for a different purpose, related, breaking down the polylactic acid we use for disposable foodware on campus. And so these two projects kind of came together through this thermophilic community. Now, I'll tell you right off, we are working under non-thermophilic conditions, but there's a possibility the enzymes are still functioning. In addition, nutrients in the microbial community. Darn it, keep doing the same thing. All right, here we go. All right, we will go back. I didn't have this problem all the years I've been doing Zoom. Just came up here. All right. We will, sorry guys, you're gonna get epilepsy watching this. It's something with this laptop now, I guess. Okay, I'll just use this button. as I said, simple stuff you can get a handle of. It's really just looking at weight loss over time, a 55-day experiment, mulch sections before and after, what is their weight loss, and then there's an indirect titration of carboxyl groups that requires a little explanation. Here's just sampling. We only did 6 replicates just because I want to test several conditions, otherwise it would become too unwieldy, but it's enough to glean some significance, but we're going to do some follow-ups and do more replicates. Okay, so there are, some of these obviously show a lot of degradation, others not as much, but there's some that show, apparent degradation, but it really doesn't show up in the weight change so much. And a couple of possible reasons for that. One thing I would point out, just something as simple as this, there's some complexity involved. So in that weathering process, there's amorphous regions that are introduced into this mulch, 28 microns thick only. And, breakages through photo oxidation and, some microbial attack. And any place where there is a breakage, that induces other breakages. And one of the reasons for that is because you form a carboxyl group that, just point to that there, that is acidic, that then enhances the rate of breakdown of future hydrolytic events. Okay, so we see, two sections that don't show weight change. One obviously though has undergone some breakdown. So what I try to establish is another measure of kind of these early events that doesn't just rely on weight change, and that's looking to see, can we measure the carboxyl group density on these sections. Just to point out, hydrolysis of these esters generates carboxylic acid groups here, and that is what is going to be titrated. What happens, in the later events, eventually plastics lose in their entanglement. As you break off pieces, they become disentangled when they get small enough. And so, that's why one reason the cleavages all don't correspond to loss of mass. It takes a while before they get small enough to diffuse away. All right. Get into that, and so that's where we did this titration, just using a pH meter. These things could be done in, all these things could easily be done in a simple classroom lab at any level, really. And just want to show you, again, focusing on this real quickly, on the x-axis here, we have weight change, and you can see there's a big grouping here that doesn't show up. Let me just, I got 5 minutes. Wow. Okay. I'm talking faster. And so we have this this large clustering here, but you can see that measuring the carboxyl group density allows you to distinguish between these. There does appear to be a upper limit, and that the way we could explain that this density reaching an upper limit is just, eventually you get these cleavages, and you do lose, for every new cleavage, you lose that carboxyl group. And what this up here in this region corresponds to, just put a best fit line there, is where you get the loss of entanglement. And what it corresponds to right around here is about, if you just theoretically, you could calculate about 1.3% of the ester linkages being cleaved will lead to this loss of entanglement. And that's right around where we are here. About 10,000 kilodalton fragments coming off. All right, so just to point out, getting back to our treatments, that looking here, we see that the most dramatic, the only significant effect we saw was of that pre-treatment, and 2 of those samples coincidentally happen to be the ones with the most weight loss and the highest degree of carboxyl groups on it. So it does appear promising, and it's worth doing some follow-up on those. And talking about experimental directions that we're going to go in. Now, what we've already started in the Sonoma State greenhouse is a follow-up just to, get some more replicates in there. In addition, include some replicates with and without nitrate to enhance a presumptive bacterial community effect. And then some other things that just kind of obvious follow-up, like are these enzymes that are in this thermophilic community still active at these mesophilic temperatures? Well, we can still denature those enzymes with enough heat, well above 63 degrees Celsius. And we also, from the mulch pieces that were degrading the fastest, we started a mesophilic community that grows just under ambient conditions and degrades mulch. And that could be something to do comparative experiments with to see, is it even necessary to work with a thermophilic community to do this? Yeah, and then also, this indirect titration method, it hasn't been used for this purpose before. It's used for a variety of purposes, but some follow-up just to refine the method a little bit. Of course, doing a time course looking at the degradation, and could be useful in the field. Instead of doing these long-term weight loss experiments, you could get a sense of early events going on and get more done that way. Now I have the slide that was requested. This one, it's a little simpler, the conclusions. What is my research about? Finding ways to speed down the breakdown of biodegradable mulch and soil. How my research impact others. Development of a simple and cheap method would enable broader use of biodegradable mulch in agriculture, lessening its reliance on polyethylene plastic. All right, so I will end with that and leave anything else for questions. Thank you for your attention, of course. Let me go to a different view here.
Anyone have any questions for Dr. Cohen? You can either raise your hand or unmute yourself.
Zoom.
I guess I have a question. This is really interesting to me as someone who grew up in the Northwest, and I saw lots of fields like this. I never understood what exactly was happening with the plastic mulch. So thank you for that explanation. How long has this has biodegradable mulch been developed? Is this a recent thing, or has this been happening for?
It's been around in the past 20 years at least, but it's catching on more. But they nothing to really even make a dent in the polyethylene plastic use. And it's just to make it easier for farmers. Yeah, if you could just till it in and not worry about accumulation, I think you'd win a lot of people over. There's a lot of inertia in agriculture. People understandably, they don't want to switch to things that cause a problem down the road. So
Thank you.
thank you.
Any other questions? Going once, going twice.
Right. Okay.
Okay, I guess we'll move on to our final presenter for the day, which will be Jim Pesavento.
All righty. Let's see here. Just gonna move this somewhere else. Okay, everybody can see my screen. Okay. All right, so today I'm going to talk about work I've been doing in collaboration with Gary Carpin, who's in the biosciences, and also just recently, 3 years ago, became a professor at Berkeley. So a lot of this work was done in the lab at Berkeley, part-time, then at St. Mary's the other part of the time. So I will be talking about top-down mass spectrometry of histone extract from staged Drosophila embryos, and I will define all of those words in the presentation so that you actually know what I'm talking about. Okay. and I'm a biochemist. So in same vein as Chris and Michael. Okay, so St. Mary's College, it's in Moraga, about 5 miles south of Walnut Creek, so right outside of the San Francisco, Oakland area. Beautiful campus, very bucolic. We have about 2,500 undergraduates or so, about 500 in School of Sciences. My research team this past year had quite a few folks. The student that I brought with me this summer is Miriam, here in the middle, in the tank top. She is working with me on a project that I'm not going to talk about this summer. I'm going to be talking about something else, but I mentored her on another project. So, and then another colleague of mine is this wonderful child. It's my son, pipetting. He's making compound P. And as you can see, full PPE, and he's loving it. So I figured Chris showed off his kid in lab, and might as well follow suit. Okay, so the overview of today's talk is I'm going to give you some background on chromatin, histones, and gene regulation. I'm going to talk about Drosophila melanogaster, or the fruit fly, and what happens during embryogenesis. The research question that I'm going to focus on for the presentation is, how do these DNA influencing proteins change during embryogenesis? We'll talk about some methodology that I use to answer this question, primarily protein extraction and mass spectrometry. And then the results I'll get into is how we identified lots of these different histone proteoforms, like different types of proteins, and clear changes that we're seeing in abundance during embryogenesis. And yeah, I wish I had another collaborator, Trent Northern. He has lots of mass spectrometers in his lab, lots of high resolution ones. Maybe I should send him an email. I don't have any mass spectrometers in my lab, unfortunately, and have to send out a lot of these samples to other places to get them analyzed. Okay, so the fruit fly annoys us all, but it's a great model system. Its genome is sequenced, so we can infer a lot of proteins. Of course, this fly is made up of tissues. Those tissues are made up of cells. Here's a nice little fluorescently labeled slide showing sort of the perimeter of a cell and then the green nuclei in it. And my research interests actually stem inside the nucleus, where I'm always interested about how DNA, which isn't naked in eukaryotic cells, but rather wrapped in proteins, how those proteins influence how DNA can be compacted into a very tight structure, how it can be opened up and different genes can be expressed. And the primary proteins that come and compact DNA and regulate whether a gene will be expressed are the histone proteins. So about 8 of them come together and they form this little ball, little scaffold, and the DNA roughly wraps around it twice to make what is called a nucleosome. So this is kind of like the first level of compaction. And then what these little beads on a string then do to be more compacted is they bind other proteins, and it eventually will fully condense the DNA into this typical mitotic chromosome during mitosis, and that's a fully condensed state. So your DNA is anywhere from, somewhat naked, like these little linker regions, to being like slightly condensed and packed with protein to being fully condensed. So so my research interests lie in understanding the types of histones that are there in the cell and what kind of chemical modifications occur on them. So here's another image of a nucleosome. Yes, they wrap DNA twice, but they have these peptide extensions that come out and can be recognized by other proteins themselves, other enzymes, and the amino acids can get modified. So you can have acetylation be added to lysines, phosphorylation, phosphate groups be added to serines, et cetera, et cetera. And these chemical modifications are reversible, and they might change the behavior of the DNA in that area. So for instance, you could have some histones that might have a methyl group on it, and that methyl group is telling the cell and the surrounding areas, okay, turn off the gene here, even though the DNA is right here, turn off the gene and not have it be expressed. Whereas you might have on the same histone in a different situation, maybe an acetyl group being added, and then that flags the area and tells the cell, okay, I want this gene to be expressed. and please express the gene, I guess. And so this is kind of a simple example of what could happen. Of course, nothing in nature is so simple. Here are the 4 histones that can be found in the nucleosome. Here's the linear sequence of amino acids, and they can be differentially modified on different sites, and in some cases heavily modified, upwards of 20, 30 modifications. Now, they can happen singularly or in combination. So the take-home message here is that it could be a very complex scenario. And so we want to analyze and study these modifications on the histones when the fruit fly develops. And that is the goal of the project. So when fruit flies are developing, they undergo embryogenesis. So here is an egg that is just fertilized. And then there's a lot of nuclear divisions. And eventually what happens is the nuclei kind of start to migrate to the outside and then cellularize, meaning that they form compartments that will become the cells. They start dividing, and then eventually this will turn into one of those wonderful little worms that crawls around your fruit before they pupate and go into flies, adult flies. Okay, so we're interested in collecting these samples at different stages in embryogenesis, the cycle 10, cycle 13, cycle 14, and early and late cycle 14. And so what we want to do, and the question that we have is, can we get enough material to extract proteins and have those proteins have sufficiently high enough histones to actually analyze by mass spectrometry? And then if the answer is yes, and we want to analyze them then, and then see if there's any differences between these different cycles. So what's shown here is called an SDS page gel. So all of these bands that you're seeing are different proteins, and proteins that are up towards the top are bigger proteins, bands towards the bottom are smaller proteins. These are my histone standards that tell me different amounts of histones and where I'm expecting them to see on the gel. And then, so if my standards are here, I'm looking for proteins and bands over in this region. Oops. And after we've done some protocols, optimizing some of the extraction methods, you can see that some work and some don't. So for instance, the cells or the embryos that were harvested at cycle 10, this is the lane that corresponds to that, not so much protein. But then we did cycle 13, and then we have 2 replicates for that. You can see there's protein bands there, quite a bit nicely in the region that we want. And then finally, let's see here, right? And then the other 2 stages also gave us some protein bands in the regions that we want. So this was promising. So what we did is we took the rest of the sample. So we're loading about 10% on these protein gels. We took the 90% and sent them to the mass spec facility to get analyzed. So time is always an issue. So how do we analyze these on the mass spectrometer? So we can take the protein extracts. We can separate them out on a column. So protein is just coming out over the course of an experiment, getting analyzed by the mass analyzer. This gives us a mass spectrum that gives you signal proportional to the mass over charge. And we're using high resolution mass spectrometers, meaning we can actually get isotopic resolution of these of the isotopes within a protein of a specific molecular weight, meaning that this species has all carbon 12s. This has 1 carbon 13, 2 carbon 13s, and of course other stable isotopes of oxygen and nitrogen contribute. But we get really high, really nice accurate mass when we analyze the proteins using this type of mass spectrometer. But so great, you have a mass, but that doesn't really tell you the identification of what the protein is. So what needs to happen is you have to have your masses. You have to actually isolate this mass in the instrument, bombard it with gas, and it'll fragment it. It'll become fragmented. And so all these fragments correspond to this one mass. And then you can kind of like a puzzle using some software, reconstruct what these fragments are, and then identify what the sequence is and where the modifications are. But if you don't know this protein a priori, you can use databases and take all this data, put it into these databases, and it'll kind of pump out and show you what proteins are likely there. So this data looked great. We were getting information on the histones from our extracts using mass spectrometry, and we were using these databases to identify them. Now, but we're not just looking at one protein. So this is a feature map that shows proteins from, say, like 10,000 to 16,000 Daltons, and where they're coming off during the time. And if we kind of zoom in, you can see more masses. And then if you zoom in even more, you can see that there's lots of clusters of these different masses. And so what we wanted to do is we wanted to see if we could use some of these database searches and then find out, okay, I see these masses, but are these masses these specific proteins? And so after database searching, we can say, aha, yes, this is indeed histone H2B. And all of these different mass increases that you see here correspond to different modifications that we've identified, specifically methylation, oxidation, things like that. So then we wanted to skip this a little bit. So then, right, we have all these data sets. We've identified some of the histone proteins, and we have all of these different conditions with thousands of mass spectra. So last summer, I spent all summer writing a Python script that takes all of these mass and abundance values, searches them for specific masses, and then outputs the mass the abundance of these different masses for your different conditions. So what you're seeing here is those 3 of the 4 different embryo cycles that I've talked about previously. And then here are the masses that are coming up. And something that was interesting to me is that we were seeing clear differences. Well, not clear. I mean, got to do this a couple more times. The error bars are pretty big, but there looked like there was differences in some of these masses as the embryo was developing, specifically on this form that is acetylated. So we took a look at other histones to see if we were seeing similar trends. And it looked like on this histone H2A and this histone H4, during embryonic development, we were seeing increase in acetylation, which was really remarkable because this seems to be right where you have this transition between expressing transcripts making proteins that are coming actually from the mother, and then eventually coming from the cell's own genomic information, which is right at about cycle 14, early and late 14. So, so why is that important? You can see this. There's an uptick in zygotic transcription right at these points, and it's known that acetylation of histones is something that correlates very strongly with increased gene expression. So, this mirrors nicely with the increase in transcripts that we're seeing. And what we need to do next is say, okay, great, we have the acetylation, we know it's increasing, but exactly where on the protein is it happening? Is it happening on lysine 5? Is it happening on lysine 16? And then once we know that, we can start to uncover the enzymes that might be playing a role in adding those acetylations during these cycle events. So the future directions that we have in mind, basically, I've just shown you sort of a little bit of the data because I only have 10 minutes. The histone post-translational modifications, there are so many of them. How much time do I want to dedicate to analyze all of this data? And my question for my collaborator is, we have a really nice workflow. We're getting really great data, but it's the big data problem, which is, okay, you can drown in it, right? We're going back and thinking about what specific questions we want to tackle with all this data, so that we're not just like, cataloging or dumping out or spending hours looking at things that might not be very interesting. So we're thinking about how to make a more targeted approach with this workflow. And then what I've shown is, so the data looks pretty good, but we could probably increase the number of proteins we identify and our confidence in abundances if we actually had more starting material. And also thinking about well, maybe we should look at what's happening to the histones outside of just the cycle 10 to cycle 14 window. What about a larva? What about an adult? What about an unfertilized egg? What are the histone modifications happening at those points? And then maybe we can try to connect it all together and build a story of how the histone proteins and the modifications are changing during embryogenesis. So that was fast and a lot, but I think I covered all the points that I wanted to cover. So I think I'm on time. So I'll take any questions that you might have. Yeah.
Thanks, Jim. Really good stuff.
Anyone have any questions for Jim?
I do.
Okay.
All right.
Yeah, thank you, Jim.
Yeah.
I know it's only 10 minutes, but maybe can you give us some background on maybe what's known about the signal transduction during this transition period, going from maternal to the endogenous genes? Are there mutants that you can work with in that pathway, or even like with the histone acetylases that exist among as a Drosophila resource?
Right. Yeah. There's quite a few mutants. So I think like the signal transduction pathways that are changing these modifications as the organism develop, I think actually they're very conserved, and a lot of these a lot of the enzymes, once you knock them out, they're lethal. So the flies just won't develop. I think one of the questions that we're starting to have now is, we're looking at global histones. We're looking at all the chromatin. So a really neat natural extension would be like, oh, can we immunoprecipitate only this acetylated group and find out what genes this acetylation mark is present at? And that might help us kind of like narrow in and find specific genes that are changing, or the chromatin environment that's changing. Mm-hmm. Because there's also like, RNA-seq data that correlate with that. But it'd be nice to tie in the modification changes we're seeing to actually localize and find what genes might be influenced, or what regions of chromatin these acetylation marks are happening in.
Hey, Jim, enjoyed the talk. Got a question about the sample enrichment.
Sure.
So it's got two parts. How well can you synchronize the larva? I used to work on yeast, and it was a real challenge to do that sometimes. And so I'm just wondering about the larva and how well you can keep them in the same stage.
Oh yeah. So that's, you have to be like monitoring when the females are laying the eggs. And you have to be really meticulous about what you're collecting. I have a great collaborator who has a postdoc that is on it. She actually will, out of a sample, she'll collect and take microscopy images to actually give me a percent. Oh, of this pool, 90% are in cycle 10 for sure. Of this pool, like 95% are cycle 13. So the staging, if you're paying attention and watching the females lay the eggs and collect them, you can get it pretty high, 90 to 95% or so.
Oh, cool.
Yeah.
And then the second part is, in a previous scientific career, I worked on histone modification in yeast as well.
All right.
And
right on.
We used to do enrichment for mass spec, and we would do Triton acid urea gels. And you can separate the histones by their modification state.
Yeah.
Is that been something you've thought about pursuing to try to, keep certain modifications together?
Yeah. So I also, back 20 years ago, have run those drills out to actually get the acetylation forms running separately to quantify them. We haven't done that because, and I didn't talk about the chromatography all that much, but these new columns that are, it's not really, whatever. The new columns do a great job at separating out the acetylated forms over time because of the change in hydrophobicity. So we actually can get information on those forms, and they're not drowned out by the more abundant unacetylated forms. And also because they're in sort of they're in the same sample, and they're on the column and kind of alluding together, we can start to quantify based on their abundances how much of these acetyl forms are there.
Any other questions for Jim? If not, we're going to give you back the rest of your time for this afternoon. I will be sending out announcements shortly to your email address. And I look forward to seeing you all again next week for our final meeting of final presentations. Have a great day.